Welcome to the Science Plus Love Equals Cure podcast, the STXBP1 10-minute, approximately, monthly update. This is podcast number 26. I'm Charlene Sunrigby, president and co-founder of the STXBP1 Foundation. I'm also mom to STXer Juno and her big brother Luca. We are just coming back from the STXBP1 Research Roundtable and Family Summit in Heidelberg, and there's lots to share from this. And so we only have two topics for today's podcast. And the first one is going to be about our ICD-10 code for STXBP1. And the second one will be an interview with Jim Goss, where we'll get a debrief on the STXBP1 Summit in Heidelberg. First topic, ICD-10 code. STXBP1 has its own ICD-10 code. So this saga was a 4-year journey that started in 2021 when our foundation applied for a code with the CDC. There has been amazing support from the community over these 4 years, including signed letters from more than 3,000 people in support of our ICD-10 request. This included more than 125 clinicians and healthcare workers. And I would think this really highlights why having an ICD-10 code is important to healthcare workers. So some of you may be wondering, why should I care about ICD-10 codes? So these codes are called International Classification of Disease codes, and they're used by healthcare providers when they see patients. They capture information in the electronic health record, and these include diagnoses, symptoms, as well as medical procedures. So without an ICD-10 code for STXBP1, the information that can be captured is information like symptoms. So seizures, developmental delay, intellectual disability, etc. But the symptoms for STXBP1 overlap with a lot of other neurodevelopmental disorders and epilepsies. So we can't use symptoms only to identify our STXers. This ICD-10 code for STXBP1 means that STXBP1 patients can be identified easily from information in their medical records, and this will enable access to better, more appropriate care today and the potential to identify patients when there are clinical trials that they could be eligible for. So it's very exciting, and our new code is QA0.0141. And you don't have to memorize it. We have flyers for you that you can download from our website. So I wanted to make sure that it's clear that the STXBP1 code does not replace other condition codes or other symptom codes. We're expecting that it will be used in addition to other codes like autism and seizures, and your provider will know how to manage this. So the key call to action is making your provider aware of this new code. As I mentioned before, we have flyers on our website that you can download and print, both in black and white and color. Okay, so looking forward, some of you may also be aware of ICD-11, which is the newest version of the International Classification of Diseases codes. So rest assured, I have already applied for an ICD-11 code, and there in most countries there are still probably 3, maybe even 10 years before it's going to be used. But when that ICD-11 version does go into use, hopefully we will have an ICD-11 code for STXBP1. Okay, second topic. I interviewed Jim Goss for today's podcast. He is our scientific director for the STXBP1 Foundation, and we were both able to attend the Research Roundtable and Family Summit in Heidelberg last week. So let's listen in to the conversation with Jim. Welcome to the Science Plus Love Equals Cure podcast.
Well, hi, Charlene. Thanks for having me.
So you and I were just in Heidelberg at the second STXBP1 summit, which included a research roundtable and a family day. And so I wanted to, use this time to ask you about it and get your impressions. So maybe what we could do is we can start out with, what's the purpose of the Research Roundtable meeting?
Well, the meeting's an opportunity for scientists and clinicians involved in STXBP1 research to come together. They share new findings and insights, and they discuss topics of importance. And the idea and the goal is increasing our understanding of all things STXBP1. The meeting is held yearly and it alternates between the United States and Europe. We're lucky that we have a number of researchers and clinicians in the US and a number of researchers and clinicians in Europe. And by alternating the sites, it gives researchers from both sides of the Atlantic the ability to be involved.
Yeah, and this was really cool that this is now the second time that we've been able to do this in Europe. We were in Milan two years ago, and I just think that it is so important for us to be creating this like this a real global community of researchers. And yeah, and in the US and Europe and beyond, it was very cool that we had one researcher, Laura Croft, who came from Australia. So I think that she gets the award for farthest travel. So,
Indeed, yeah, we usually have somebody from Australia or a couple of people from Australia last year when we had the meeting in the US. So,
yeah.
yeah, they often get the award for traveling the furthest.
Yes. Well, what were I'd love to hear about your impressions from the research roundtable. Like, what were your top, say, three takeaways from the meeting?
Yeah, it's a great question. Well, first, before I get into the takeaways, it's just I want to how amazing it is to see the advancements that have been made in STXBP1 field every year. Every year we get a deeper understanding of the pathophysiology, the progress therapeutic development for STXBP1, and improved approaches in monitoring the disorder's clinical progress. And we had talks on all of those subjects at this year's roundtable. But I guess if I had to choose three kind of key takeaways from the meeting, I would say that there were a couple of research challenges that were discussed that I think are going to require more focus moving forward in the future. And there's one challenge where it's clear that we've made a tremendous amount of progress. And so the first takeaway, which is one of the research challenges, are questions concerning STXBP1 gene variants. There are still some questions involved in these variants that we don't really have a complete handle on yet. And so probably everybody understands that most disease-causing STXBP1 variants result in either no protein being made in a neuron or the formation of a non-functional protein. And that's what leads to the haploinsufficiency that causes the disorder. But there are some missense variants that might produce a mutant protein, a mutant STXBP1 protein that could interfere with the normal STXBP1 protein made by the normal gene. And if this happens, this is a phenomenon called a dominant negative effect. And there were a couple of talks at the research roundtable that were investigating the potential of missense variants to cause these dominant negative proteins to be made. And the evidence presented at the meeting suggested that this is not the case for the variants that were investigated. However, there's still no definitive evidence to confirm or exclude the possibility that these types of proteins will be made. And the reason why this is important is because there are types of therapies, so certain antisense oligonucleotide or ASO approaches, or CRISPR-A approach of therapies, which are designed to interact with the person's own genes to increase protein production. And so if you had a dominant negative protein, then if you were increasing the production of that, it would just interfere with the protein, the normal protein you're trying to increase. And so those therapies may not be appropriate for individuals who have those variants. So this is an important question for us to better understand. We're making progress, but we need to do more work with that. And so that kind of leads me to the second takeaway, which is the fact that more needs to be done in therapy development. We've made great strides over the years, but more does need to be done. Obviously, what happened in the capsida trial was devastating. That particular therapy involved gene replacement or the addition of an exogenous STXBP1 gene into neurons. It used a unique AAV viral vector. We don't know yet what went wrong with that particular trial, but there's going to be a lot of research and a lot of time that's going to be needed to figure that out before that can move forward. Uh, but other approaches, such as the ASOs and the CRISPR-A I just mentioned, were topics of a few talks at the Research Roundtable. And as I said, these therapies are designed to increase the amount of protein made from a person's own genes. And so far, what investigators have found is using these approaches, they've been able to increase the amount of protein about 50% in iPSC derived neurons. And so obviously in a haploinsufficiency disorder where the amount of protein that is being expressed is half, you would like to double that. And so what we do see is we see about a 50% increase. The issue is we don't know what that's going to do in a patient. So we don't know if 50% is enough or if you're going to need to make more than 50%. And really the only way to address that question is to have a animal model where you can test the therapy in and see if it has any effect on phenotypes, so seizures or motor issues or what have you, but you need an animal to do that. And because these are therapies that have to interact with the specific human sequence, you need what's called a humanized mouse, which is basically a mouse where you put in the human STXBP1 gene. So if you have a mouse like that, you can test that. So attempts have been made, or attempt was made to make a humanized STXBP1 mouse. That mouse was created, but it isn't fully functional. So some questions can be addressed with that mouse, but not kind of this main question of is 50% protein enough? And so work on trying to develop a humanized mouse is something that needs to continue because we really need to answer that question. How much protein do you need to increase in the neurons in order to have an effect, to make an effective therapy?
Right, because these ASOs, like different from a gene therapy, these ASOs are actually interacting with the existing gene, right?
Right.
And so that's why you need the humanized gene in the mouse model versus being able to just work with one of the existing mouse models that we've had.
Exactly. And I also want to say that these types of therapies, these ASO therapies and CRISPR therapies, and these types of therapies that interact with the person's own genes, these are technically difficult therapies to develop. There's a lot of trial and error with trying to make particular ASOs and what parts of the gene you want to interact with or parts of the RNA that you want to interact with. And so these studies take a long time and it is just very difficult to perfect the actual therapeutic product that's eventually going to come out of that. So while the first two takeaways were kind of research challenges that are going to require more attention, moving forward in the future, I think my third takeaway is a challenge that we are definitely meeting, and that is that we are absolutely crushing our clinical trial readiness. And so there were a few talks on natural history studies uh, and the outcome measures that are being collected. And it's clear that we are collecting very valuable data in our STAR natural history study. And the outcome assessments that we are collecting are looking increasingly promising, that they will be able to measure meaningful changes in upcoming drug interventional trials. And that's really the purpose of the natural history study. And importantly, pharmaceutical companies are noticing the work that we're doing and praising our efforts in our natural history study. There were also a couple of good talks on EEGs and EEG analysis. And these are going to provide, I think, further evidence uh, on how important EEGs will be as biomarkers moving forward. And in addition, I also point out that the ESCO natural history trial will be starting soon. And what's great about that is we'll be collecting natural history data from not only the US, but now we're going to be collecting it from Europe. And that just makes STX-BP1 even a more attractive disease indication for pharma to develop therapies for.
Wow. So a lot covered in the 2.5 days of the research roundtable, and I guess that it's no surprise that science isn't a straightforward path, but it's also really great to hear that there's a lot of work happening, and also that the natural history study efforts have been really going in the right direction. So I'm curious, was there one insight where you were surprised, coming out of the meeting?
Yeah, so Francisca Furia from Denmark gave a talk on mild cases of STXBP1, and it was a very interesting talk. And so these are individuals who have STXBP1 variants, but they can walk and they can talk, although generally their ability to walk and talk was delayed from what you would see in a normal developing a child. They have a bit more than half of these individuals have normal cognitive abilities and no significant neurological abnormalities. Most of the patients have very well-controlled epilepsy, but the majority suffer from autism or some sort of behavioral disturbance. And so this group of individuals, and I forget exactly the number of individuals it was, I want to say 16 or 20 or something, but it was not a small number.
Hmm.
Uh, but it'd be great to be able to study them in a bit more detail, and ask some questions, like how do their EEGs differ from what we see in our typical STXBP1 individuals? What's different about the variants that they have that we don't see in your more typical STXer? Do they have true haploinsufficiency? Uh, and or is it just a partial reduction in STXBP1? And are these the types of patients who are what how they present? Is that representative of what we might expect a successful therapy to look like if we treated a typical STXer?
Wow, that's really interesting, and really expanding our understanding of the spectrum
Yes.
of
Yeah.
what STXBP1 can look like in individuals. Very, very interesting. So, coming out of the research roundtable, what do we do with all of this information? So, what do you see that you and other researchers are going to be able to do with the insights and the learnings from the research roundtable?
Yeah, well, the purpose of these meetings obviously are to provide researchers with opportunities to share the information, things that they've learned in their own lab, but they can share it with others. And hopefully, researchers will take away from the meeting ideas that will help them refine their own work, or maybe they'll come away with new ideas to start new projects. We always hope that they come away with some collaborations between researchers who may not have been collaborating before, but now kind of found common ground to say, hey, maybe we should work together on a particular project. And this kind of collaboration and discussions occur not only because we have the formal presentations during the meeting, but it's really the casual conversations between researchers at like coffee breaks and lunches and dinners. That's where the real meat and potatoes of collaboration and discussions take place. And researchers from different countries and different areas, they all run into some of the same common challenges whenever they're doing research. And by basically discussing amongst themselves, they may find new avenues to address some of the challenges that are just present, or that they've encountered. So.
Yeah, yeah, I thought it was really there was a section of having having researchers from other diseases present different studies that hopefully we can learn from. And I thought that there was a really cool interchange around what we can learn, from, and in this case it was Dravet syndrome. But then this whole idea that actually taking that forward, are there ways that we could work together? Across the disorders, and even potentially, kind of across DEEs, developmental and epileptic encephalopathies. So I think it's really hard, even though virtual meetings are, still so valuable and critical to keep the momentum going. I think it's still really hard to replace that in-person interaction.
Yeah, absolutely agree. I mean, these kind of meetings are absolutely vital. And you brought up an excellent point about learning from other disorders, right? So, the other DEEs that may have had the same questions that we're trying to struggle with may have answered those, or may have come up with a kind of a unique way of addressing those questions. And obviously that's something we can bring into the STXBP1 field.
Yeah, yeah. Well, so how is this going to affect your work and how you're looking at prioritizing things going forward?
Yeah, so the kind of the key questions that were raised during the meeting, and we talked about a couple of them, whether STXBP1 variants exhibit dominant negative phenotypes, the degree to which you have to increase STXBP1 protein to have a therapeutic effect. There was also questions brought up about what's the potential influence of STXBP1 haploinsufficiency in prenatal development, and how do we ensure that we're collecting the best clinical data, in our natural history studies so that we optimize success for future drug interventional trials. So those kind of questions that are brought up during the researcher roundtable are questions that we will, as a foundation, start to focus on, and that we will be working closely with our scientific advisory board as we move forward to execute our new 3-year strategic plan for the future. So,
And you know how I love plants, so that pleases me. Well, just to round things out for the summit, what gave you hope?
it's the people, right? So it's the researchers, the clinicians, the healthcare professionals, the families, the farmer representatives who were all present at the meeting, all came together with a single purpose, and that's finding a cure for STXBP1. And they are all, to an individual, determined and driven, and I can't help but believe that there will be significant progress moving forward in the future.
Yeah, that's great. I also came out, pretty excited about, there's clearly a lot to do, but also seeing the amount of progress that's happened in the last year. And you're right that it is really through bringing together, researchers, clinicians, patients, industry that we get the right cross-pollination, to drive things forward. So, um, well, I have one more question, but I want to kind of take a little bit of pivot to another topic. So, in the month of October, there have been a number of grants that the STXBP1 Foundation has announced, that are being given out, and so can you talk a little bit about the significance of those grants, and how are they aligned with what our roadmap is, or how do they help us to achieve what's on our roadmap?
Sure, yeah, absolutely. So we've been lucky this year to be able to fund several innovation awards and expand the STAR Natural History Study. And so we funded two innovation awards aimed at developing precision therapies to treat STXBP1. So one of the awards is developing a gene replacement therapy, and another of the awards is developing a CRISPR-A therapy. And while similar types of therapies are already being developed, so people will ask, well, why are we funding something that other people are already funding? But that's because it still reflects our commitment as a foundation to accelerate the development of a cure because we need to maximize and multiply the number of therapeutics in development. More shots on goal is very important, and even duplicating effort um, is very, very important because you never know what therapies are going to be moved forward and which therapies are going to be left behind and which therapies are going to fail. So it's not uncommon if for example, a pharmaceutical company or even a researcher might be developing a therapy, but for whatever reason decides that they don't want to continue with that particular research. And
Mhm.
so it may still be promising, but if they're not willing to continue with that, then we lose that opportunity. And so as part of what we want to do as a foundation is to try to minimize that risk. And so those are why we fund therapeutic development projects that may be similar to the therapies which are already under development.
Right, right. So
Uh,
it's a little bit of de-risking and if you will, allowing the us to own our own destiny to some
yeah,
extent, but it
yeah,
is that there is this significant drive around de-risking because science might not, work in some of the, in some of the projects, including a project that we might fund, right?
yeah.
And it's important that we continue to accelerate these therapies as quickly as possible. And then, as you mentioned, I mean, sometimes business decisions are made that to deprioritize or not go forward with a therapy. So
Yeah, exactly. And then we funded a innovation award looking at the role that STXBP1 might play in respiratory physiology, which may seem again kind of out there, but many of our kids have respiratory issues. And in the paper that was published last year looking at leading causes of mortality in STXBP1, respiratory issues was close to the top. And so we funded a innovation award looking to see, does STX-EP1 actually play a role in respiratory function? And then we also funded a award looking at repurposed drugs to treat motor behavior or motor issues in STX-BP1. And uh, what's interesting about that is if that particular grant is successful and a drug is identified that might be utilized to treat motor problems, that's a drug that could be utilized in our kids now. because these are approved drugs by the FDA. So there's no further development needed. They can just be given. And so that's great. And then we've also been able to continue to fund several of our STAR clinical sites, and we will be funding the new site that's going to opening in Chicago. So it's been a good
Oh.
year. Yeah,
Yes, very exciting about that. And I mean, I guess that with the repurpose, so that is very exciting about the Midwestern Star site. I think that there's been a lot of desire from our families to have a site that would be closer, to home, especially since the Midwest is a really big expanse and we're going from basically Philadelphia to Denver in terms of there not being a clinic in the upper part of the US, so that's great. And I guess the other thing in terms of the repurposed drugs, that it would be pretty easy to move that into a small clinical trial in humans, because this is in zebrafish, if I remember correctly, right?
it is. Yes.
Okay.
Yeah.
That's great. That's great. Well, a lot of exciting stuff for the end of the year. Jim, thank you so much for joining the Science Plus Love Equals Cure podcast.
Oh, well, thanks for asking me on. Enjoyed it.
All right. Thank you. We'll see you all next month.
Bye.